Assalamu alaikum. Today we are going to talk about the formation and activation of osteoblasts, the bone forming cells, and osteoclasts, the bone resorbing cells. In the last lecture, we talked about the osteocytes, which are the mature bone cells or the cells entrapped within the bone matrix. And then we talked about the osteoclasts, which are the bone resorbing cells. We talked about their structure and how they resorb the bone surface. I briefly mentioned the sequence of resorptive events that occur as a result of the attachment of osteoclasts to the bone surface that they have to mineralize. And if you remember, I said that these osteoclasts, whenever they have to resorb a bone surface, that particular bone surface throws itself into a concavity called the Howship's lacuna, into which the osteoclast fixes itself, attaches there by the help of the sealing zone, and creates a micro environment between its ruffled border and the mineralized bone surface that it has to resorb. The ruffled border is that zone of the osteoclast where all of the resorptive activity occurs. Within this ruffled border, within the membrane of this ruffled border, we have the hydrogen pumps that pump the hydrogen from the ruffled border into the compartment towards the mineralized bone surface, and those hydrogen ions create an acidic environment within the resorbing bone surface, and they resorb the organic matrix of they resorb the mineral of the bone. And as a result of that resorption, basically the organic matrix of the bone is exposed. Once the organic matrix has been exposed, the same ruffled border exports or exocytoses certain enzymes like lysozymes and acid phosphatases onto the resorbing bone surface that degrades the organic matrix of the resorbing bone. Once the organic matrix has been degraded, the degraded organic matrix or organic particles are packed into vesicles, and they are taken back by the osteoclast by the process called endocytosis, and they cross the cytoplasm of the osteoclast and exit the osteoclast, as you can see here through these arrows, from the opposite border of the osteoclast, and this process is called the transcytosis of the vesicle. And at the opposite border, those vesicles, the degraded molecules, basically, they exit the osteoclast, and that is how the resorption of the bone was happening by the osteoclasts. In today's lecture, we are going to talk about basically the regulation of the formation of these bone cells, both the osteoblasts and the osteoclasts. The bone forming cells, or the osteoblasts, basically have a mesenchymal origin, as we know, whereas the osteoclasts have a hematopoietic origin. Whether osteoblast or osteoclast, both the cell types differentiate by a multi-step process, and this process is stimulated by many growth factors, hormones and cytokines, and all of this cytokine, growth factor and hormone set of molecules, they are a part of a complex pathway which is called the signal transduction pathway, where cell to cell signaling is happening, molecule to cell signaling is happening, that ultimately results in the differentiation and maturation of either an osteoblast or an osteoclast. So an osteoblast would basically originate or differentiate from a mesenchymal cell. or a mesenchymal progenitor, you could say, whereas an osteoclast would differentiate from a hematopoietic progenitor. And both these processes of either osteoclast or osteoblast differentiation, they are stimulated by the certain hormones, growth factors and cytokines. So from a basic stem cell, the intermediate progenitor cells form, leading to many post-mitotic osteoblasts, and these forms include the osteoprogenitors, which are both mature and immature forms of the osteoprogenitors, and the osteoprogenitors basically give rise to the pre-osteoblasts, and then the pre-osteoblasts differentiate into the osteoblast proper through about 8 cell divisions. So the osteoblastic cell that we get, the activated differentiated osteoblastic cell that we get, is coming from a single stem cell. From a single stem cell, the progenitor cells form, and these progenitors differentiate into osteoprogenitors. The osteoprogenitors could be mature or immature. The osteoprogenitors mature, then give rise to pre-osteoblasts, and then the osteoblasts pass through certain stages of differentiation to develop into the osteoblast. This figure here briefly summarizes the formation of an osteoblastic cell. As I have mentioned, that the formation of an osteoblastic cell is a signal transduction pathway that involves many growth factors, cytokines, and hormones. And we know that the osteoblasts are coming from the mesenchymal cells. So the stem cell would be mesenchymal in origin, as you can see here in this figure. This mesenchymal stem cell has the capacity to give rise to either a cartilage forming cell, or a muscle forming cell, or a fat forming cell, or a bone forming cell. So the single progenitor could differentiate into these 4 types of cells depending upon the stimuli. And what are those stimuli in case of the osteoblast differentiation? They are signaling molecules, transcription factors, growth factors, certain hormones. So the mesenchymal stem cell could give rise to a cartilage forming cell through the formation of the prechondrogenic cell. It could differentiate into a myogenic cell, or it could differentiate into an adipocytic cell, or it could differentiate into an pre-osteoblast. Now when we need the pre-osteoblast, basically pre-osteoblast is a precursor for the osteoblast. So when we need the osteoblast, we need to push this differentiation of mesenchymal cell towards the pre-osteoblast differentiation side, and we have to block this where we need the osteoblasts. There are certain factors that halt the differentiation of mesenchymal cells into the prechondrogenic, myogenic, or the adipocytic lineages, and direct the differentiation of mesenchymal cells towards the pre-osteoblast formation. Then we have certain transcription factors like RUNX2, the runt-related transcription factor 2, Osterix, and beta-catenin, which basically are involved in the formation. RUNX2 is involved in the differentiation of pre-osteoblast from the mesenchymal stem cell, and then the Osterix and the beta-catenin, they direct the pre-osteoblast differentiation into the osteoblasts. So let's have a brief recap of what a transcription factor and a signaling molecule was. Transcription factors basically are certain proteins that are involved in the process of converting or transcribing the DNA into RNA. And signaling molecules are those substances which are synthesized by cells for the purpose of extracellular communication between the cells. So these signaling molecules basically cells synthesize them, and these molecules basically signal these cells and the cells around them towards a certain pathway. So transcription factors basically are proteins that are involved in the conversion of DNA into RNA, and signaling molecules are substances that basically are involved in the extracellular communication between the cells. And these two molecules, transcription factors and the signaling molecules, have a very important role in the formation of the osteoblasts. Okay. So as I mentioned that we have a very important transcription factor RUNX2, the runt related transcription factor 2, which basically pushes the mesenchymal or allows the mesenchymal stem cell to differentiate into a pre-osteoblast. And then we have another type of transcription factor, the osterex and the beta-catenin, that differentiates the osteoblast from the pre-osteoblast. Now let's get into the details of what these transcription factors do and how they regulate the formation of the bone forming cells. Talking about the runt related transcription factor 2, this transcription factor basically is a master regulatory switch which mediates both the activation and repression of the cell growth and phenotypic genes as the osteoblasts progress through different stages of differentiation. Also, this transcription factor triggers the expression of many major bone matrix proteins like sialoprotein, osteocalcin, type 1 collagen, osteopontin, and they control the maturation of osteoblasts and their transition into osteocytes. So this runt related transcription factor 2 basically is involved at every step of differentiation of an osteoblast. First, it activates and represses the cell growth as the osteoblast progresses through different stages of differentiation from the mesenchymal stem cell to the pre-osteoblast and then to the osteoblast, and even from osteoblast into an osteocyte. This runt related transcription factor basically is involved in all of these steps, and along with that it also triggers the expression of many bone matrix proteins like osteopontin, osteocalcin, type 1 collagen, and bone sialoprotein. It also controls the maturation of an osteoblast and conversion of an osteoblast into an osteocyte. So it is involved basically at almost every step of the differentiation of an osteoblast, from the stem cell to the pre-osteoblast to the osteoblast, and then from osteoblast to the osteocyte even. At all these steps, this transcription factor has a very important role. However, there are other transcription factors like runt-related transcription factor 1. These transcription factors allow the stem cell or push the mesenchymal stem cell to differentiate into other lineage of the other types of lineage like chondroblasts or adipocytes or the myogenic cells. So transcription factor 2 basically is pushing the formation of the osteoblast from the stem progenitor cell. Then we have another transcription factor by the name of osterex. This also plays a very important role in directing the precursor cell away from the chondrocytic lineage towards the osteoblastic lineage. So basically, as I said, that RANKL is pushing the precursor cell or directing the precursor cell towards the formation of an osteoblast and not towards the formation of other types of cells. And the RUN related transcription factor 1, for example, directs the formation of other types of cells from the precursors, like the chondrocytes or the myogenic cells or the adipocytes. Whereas another transcription factor by the name of osteryx has a very important role in directing the precursor cell away from the chondrocytic lineage toward the ऑस्टियोब्लास्टिक लीनिएज. इस बात का क्या मतलब है कि ऑस्टेरिक्स जो है वो प्रीकर्सर सेल को कॉन्ड्रोसाइट के अंदर डिफरेंशिएट होने नहीं देता, उसको वहाँ से उस डिफरेंशिएशन से हॉल्ट कर देता है और वो उसको पुश करता है टुवर्ड्स डिफरेंशिएटिंग इनटू एन ऑस्टियोब्लास्टिक लीनिएज. So Osteryx has a very important role in that. Then we have other transcription factors like beta catenin pathway and BMP2 signaling molecules that are involved in the differentiation of an osteoblast from a pre-osteoblast. Then we have various bone specific transcription factors that have also been demonstrated to affect the differentiation of an osteoblast and also the function of an osteoblast, and these include the DLX families and the genes from the MSX families. So basically all of these transcription factors and growth factors are involved in the formation and differentiation of an osteoblast from the precursor cell, which is a mesenchymal stem cell. So the mesenchymal stem cell could differentiate into a chondrocytic cell, an osteoblastic cell, or a myogenic cell, or an adipocytic cell. But what causes or what forces it to differentiate into an osteoblastic cell is the RUNX2. The RUNX2 basically controls the differentiation of the stem cell into the towards the osteoblastic lineage. Whereas at the same time, another transcription factor, the osteryx, directs the precursor away from the chondrocytic lineage towards the osteoblastic lineage. So all of these transcription factors basically are acting together. They are working simultaneously for the differentiation of an osteoblast from the precursor stem cell. Then we have certain miscellaneous transcription factors like beta catenin and BMP2s, two signaling molecules that basically regulate the formation of an osteoblast from the precursor, the pre-osteoblast. Then various bone specific transcription factors are also involved, including the members from the DLX genes family and MSX gene family that affect the osteoblast differentiation and function. So that is how the osteoblast is being formed from the mesenchymal stem cell. A lot of transcription and the signaling molecules are involved in the process, and the important ones to remember are RUNX2, osteryx, beta catenin, and BMP2 signaling molecules. This is all about the formation of an osteoblast. Now we are going to talk about how we get a mature osteoclast from the stem cell. As we know that osteoclasts have a hematopoietic origin. They are multinucleated cells. They arise from hematopoietic precursors of the monocyte macrophage lineage. So that makes their origin different from the origin of the osteoblasts. All of the cells of the connective tissue, the stromal cells basically within the marrow cavity, as well as the osteoblasts, modulate the differentiation of osteoclast via two ways: secreted molecules or direct cell to cell interaction. So the osteoclasts are coming from the stromal cells within the marrow cavity, and they're also differentiating from the osteoblasts. And this differentiation of the osteoclast from either the stromal cells or the osteoblasts occurs either by the secretion of certain molecules that helps in the differentiation of a stromal cell into an osteoclast, or by the direct cell to cell interaction with of the stromal cells with the hematopoietic precursor cell that results in the differentiation and activation of an osteoclast. So it could be either via secretion of certain molecules or by direct cell to cell interactions. The stem cells within the PDL are found in around the blood vessels, which we call the perivascular location, or they may be found within the bone marrow itself. The signaling pathway implicating the receptor activated nuclear transcription factor KB, which is basically RANK, and its ligand, they play a major role in the differentiation of an osteoclast, and the process of the differentiation of an osteoclast is called osteoclastogenesis. The major signaling pathway that controls osteoclastogenesis is the interaction between a molecule called RANK, which stands for receptor activated nuclear factor, with its ligand called the RANKL, the receptor activated nuclear factor ligand, RANKL. So interaction between these two molecules, the molecule RANK and its ligand, it plays a very important role in the formation of an osteoclast, and that process of the osteoclast formation is called osteoclastogenesis. So basically this RANK, the receptor activated nuclear factor, it is present or it is expressed on the plasma membrane of both the osteoblasts and stromal cells of the connective tissue. So imagine the stromal cells of the connective tissue. Let me draw here. Let me get a pointer. Let me change the color of the pointer. Okay. So the osteoblastic cell and a stromal cell, both of them, they on their plasma membrane have a factor, or you could say a molecule called the basically a receptor on their surface called the RANK. Let me label it here. I'm sorry, I have to use a different color highlighter. Okay, so this is RANKL. This RANKL is present on the surface of both the osteoblast and a stromal cell. I am writing an S for the stromal cell. So basically, on the plasma membrane of osteoblast and stromal cells, there is a ligand present called the RANKL, which is a receptor activated nuclear factor ligand. ठीक है, तो ऑस्टियोब्लास्ट और स्ट्रोमल सेल्स की प्लाज्मा मेम्ब्रेन के ऊपर एक लाइगेंड होता है कॉल्ड दी RANKL. This ligand is supposed to bind to a molecule called RANK, the receptor activated nuclear factor KB. This rank is present on the plasma membrane of the progenitor cells of the osteoclast. So when the progenitor cells of the osteoclast, the rank binds to this rank L on the osteoblast or the stromal cell, a system or a cascade of signaling establishes, leading to the differentiation and fusion of osteoclast precursor cells and promotion of the survival and activity of a mature osteoclast. So basically, for osteoclastogenesis or the activation, differentiation of an osteoclast, we need the interaction between rank L and rank. Rank is present on the plasma membrane of the osteoclast progenitors, whereas its ligand, the rank L, is present on the plasma membrane of the stromal cells and osteoblastic cells. So when these ligand and the molecule interact with each other or bind with each other, that results into a cascade activation of a system of pathway or a cascade pathway that leads to differentiation and fusion of the osteoclast precursor cells and formation of the mature osteoclasts. So if I show you this whole thing by the figure, this is where you have the precursor cell for the osteoclast. So let's start from here. The osteoclasts differentiate, or they're coming from the hematopoietic stem cells. So the hematopoietic stem cell differentiates into the osteoclastic progenitor under the action of a signaling molecule called the monoclonal stimulating factor, the MCSF. Once you get the osteoclast progenitor, that osteoclast progenitor differentiates into a pre-osteoclast. The pre-osteoclast has a signaling molecule on its surface called the RANK, which is the receptor activated nuclear factor KB. This RANK is only activated by the attachment of the ligand, the rank L ligand, and that rank L ligand is present on the surface of two cells, the osteoblasts and the stromal cells. So when the osteoblasts or the stromal cells, they bind to these pre-osteoclasts through their rank L, the pre-osteoclasts differentiate into the osteoclast. So for the osteoclast differentiation or the formation of a mature osteoclast, we need the interaction between these two molecules, the rank and the rank L. जब ये दोनों molecules एक दूसरे के साथ interact करते हैं तो हमारे पास एक signal क्या कहते हैं cascade of pathway activate हो जाता है जो eventually pre-osteoclast को osteoclast में, mature osteoclast में differentiate करा देता है. So this type of activation is basically the cell to cell interaction. दोनों cell की membranes basically through the molecule and the ligand interact कर रही हैं and that results in the formation of an osteoclast. अब इस activation को control करने के लिए osteoblasts एक decoy molecule भी secrete करते हैं and that decoy molecule is soluble and it is called osteoprotegerin. Osteoprotegerin basically काम क्या करता है कि ये rank L को rank के साथ interact नहीं करने देता. It basically blocks the interaction between rank L on the osteoblasts and the stromal cells and rank on the osteoclast progenitor cells or the pre-osteoclast. जब ये दोनों एक दूसरे के साथ interact नहीं करेंगे, obviously cell to cell interaction complete नहीं होगी and there would be no activation or no formation of osteoclast, and we say that this molecule, the osteoprotegerin, interferes with the osteoclast formation. So this is how the naturally the balance between bone formation and bone resorption is maintained. Whenever bone resorption is required, the rank and rank L interact with each other and the osteoclasts are produced. Mature osteoclasts are produced, and what do? They resorb the bone. And whenever bone resorption is not needed, the osteoprotegerin inhibits the binding of rank L to rank, and that in turn interferes the osteoclast formation, and that results in no bone resorption. So that is how at all times a balance between bone resorption and bone formation is maintained by basically the osteoprotegerin and the rank L of the osteoblast itself, because rank L भी osteoblast के ऊपर present होता है और osteoprotegerin भी osteoblast ही secrete करता है, तो वही ये दोनों processes को majorly control कर रहा होता है. Okay, the fusion of mononuclear osteoclast into multinucleated osteoclast and their subsequent activation is also driven by this rank L and rank system. मेजर जो डिफ्रेंसिएशन थी प्री ऑस्टियोब्लास्ट से प्री ऑस्टियोक्लास्ट से ऑस्टियोक्लास्ट मैच्योर के अंदर, दैट वाज being controlled by the cell to cell interaction through the rank and rank L system. Also, a mononucleated single osteoclast conversion into a multinucleated osteoclast is also controlled by this same system. यही rank L जो है वो mononuclear osteoclast को multinucleated osteoclast के अंदर convert करते हैं और फिर उसकी subsequent activation भी करते हैं. So this cell to cell interaction which basically is controlled by the rank and rank L system is a major factor in controlling the activation and formation of the osteoclasts. Initially, these osteoclasts that form as a result of the rank and rank L system activation, they are non-polarized cells, and they only attach to the bone by the help of certain transmembrane receptors called the integrins. But as the osteoclasts differentiate or go through the process of differentiation, they eventually become polarized cells and they develop the ruffled border, as I mentioned, that these osteoclasts have. Initially they are non-polarized cells और वो bone के साथ सिर्फ integrins की help से attach करते हैं, but then eventually they become the typical osteoclasts, which are polarized cells and having a ruffled border and sorry, let me get back. Okay, and also having the typical features of an osteoclast, which is the सीलिंग जोन. ठीक है, तो इनिशियली जब ऑस्टियोक्लास्ट डिफरेंशिएट होता है तो पहले वो मोनोन्यूक्लियर सेल होता है, फिर वो मल्टीन्यूक्लिएटेड सेल में कन्वर्ट होता है. मल्टीन्यूक्लिएटेड सेल इनिशियली नॉन पोलराइज्ड होता है और वो बोन के साथ थ्रू इंटेग्रिन्स अटैच करता है, बट देन इवेंचुअली द ऑस्टियोक्लास्ट बिकम्स पोलराइज्ड और वो रफल बॉर्डर अपना इस्टेब्लिश कर लेता है, सीलिंग जोन इस्टेब्लिश कर लेता है, एंड देन it develops all the systems that it needs for successfully demineralizing and degrading the bone matrix. So this is how you get an osteoclast from a single hematopoietic stem cell. Hematopoietic stem cell converts into or differentiates into an osteoclast progenitor. Osteoclast progenitor differentiates into a pre-osteoclast. The pre-osteoclasts have a receptor on their membrane called the rank, which is a signaling molecule. rank के लिए एक ligand होता है the rank L, जो कि प्लाज्मा मेम्ब्रेन ऑफ ऑस्टियोब्लास्ट और स्ट्रोमल सेल्स के ऊपर होता है. जब ये दोनों एक दूसरे के साथ इंटरैक्ट करते हैं, that results in the activation of the osteoclast. Initially the osteoclast is a mononuclear cell, फिर वो fuse करके by the help of this rank and rank L system they fuse together and they result in the formation of a multinucleated active osteoclast. This multinucleated active osteoclast is initially it does not have a ruffled border. It is a non-polarized cell, और ये bone के साथ सिर्फ integrins की help से attach करता है. But eventually, by the help of this RANK-RANKL system, the active osteoblast develops, which is polarized, having a ruffled border, and all of the systems that it needs for the demineralization of the bone develop within the active osteoclast, and that is how you get an active osteoclast from the stem cell. फिर मैंने बताया था कि RANK-RANKL सिस्टम को कंट्रोल करने के लिए भी एक मॉलिक्यूल है जो ऑस्टियोब्लास्ट सिक्रीट करते हैं, एंड दैट इज osteoprotegerin. This osteoprotegerin controls the or inhibits the interaction between RANK and RANKL, and thus it inhibits the activation of an osteoclast. तो जब भी बोन रिजॉर्प्शन नहीं करानी होती, osteoprotegerin RANK-RANKL का इंटरेक्शन नहीं होने देता और osteoclast डिफ्रेंशिएशन नहीं कंप्लीट होती. एंड व्हेनेवर बोन रिजॉर्प्शन इज नीडेड तो RANK-RANKL के साथ इंटरैक्ट करता है एंड यू गेट एन एक्टिव osteoclast. So that is how the two major bone cells, the bone forming cell and the bone resorbing cells, are formed from their progenitor cells. Then I'm going to briefly tell you about the development of bone. जैसे कि इस चैप्टर के शुरू में मैंने आपको बताया था कि बोन को हम on the basis of development भी classify करते हैं into 3 types: the endochondral bone, the intramembranous bone if you remember, and the third type was the sutural bone. Bone development basically is the process of the formation of bone. Histologically endochondral intramembranous bone types जो हैं उनके अंदर कोई difference नहीं होता, but the process by which they develop differs, इसलिए हम इनको एक दूसरे से अलग क्लासिफाई करते हैं on the basis of the types of development or on the basis of the process of the development. Bone is divided into or classified into 3 types: the endochondral bone, the intramembranous bone, and the sutural bone. The endochondral bone is that type of bone formation or bone development where first a cartilage model develops within the mesenchyme, then that cartilage model is replaced into a bone. That is why this type of bone formation is called the endochondral bone formation. Endo is for within, chondral is for cartilage. तो cartilage के अंदर bone बनती है. पहले एक cartilage model बनता है mesenchyme में, वो model eventually bone से replace होता है. Whereas within the intramembranous bone formation type, bone formation डायरेक्टली mesenchyme के अंदर होना शुरू हो जाती है. इंट्रामेम्ब्रेनस मतलब इंट्रा विद इन, मेम्ब्रेनस मेम्ब्रेन और दी mesenchyme. तो डायरेक्टली mesenchyme के अंदर जो बोन फॉर्मेशन होना शुरू हो जाती है विदाउट द फॉर्मेशन ऑफ अ कार्टिलेज मॉडल इज कॉल्ड द इंट्रामेम्ब्रेनस बोन फॉर्मेशन, एंड द टाइप वेयर यू गेट अ कार्टिलेज मॉडल फर्स्ट which is replaced by bone eventually is called the endochondral bone type. एंडोकॉन्ड्रल बोन फॉर्मेशन हमारी लॉन्ग बोन्स की एक्सट्रीमिटीज पे होती है, वर्टिब्राज में होती है, रिब्स में होती है, बेस ऑफ द स्कल में होती है, आर्टिकुलर एक्सट्रीमिटीज ऑफ द मैंडिबल पे होती है. सो दीज आर द वेरियस बोन्स दैट एक्चुअली डेवलप्ड बाय दिस प्रोसेस ऑफ बोन फॉर्मेशन. एंड एज आई सेड कि पहले mesenchyme के अंदर एक कंडेंसेशन होती है, वहाँ पे कार्टिलेज सेल्स आते हैं, कार्टिलेज फॉर्मिंग सेल्स आते हैं विच आर द कॉन्ड्रोब्लास्ट्स. वो कार्टिलेज फॉर्मिंग सेल्स mesenchymal सेल्स को ब्लास्ट में कन्वर्ट करते हैं. कॉन्ड्रोब्लास्ट्स क्योंकि कार्टिलेज फॉर्मिंग सेल्स हैं, दे स्टार्ट फॉर्मिंग दी कार्टिलेज. कार्टिलेज का एक पेरिफ्री बनता है, कार्टिलेज कवरिंग और कार्टिलेज बाउंड्री बनती है विच इज कॉल्ड द पेरिकॉन्ड्रियम, एंड दैट इवेंचुअली गिव्स राइज टू अ मॉडल ऑफ कार्टिलेज this model of cartilage, once you get this model of cartilage, it then is eventually आहिस्ता आहिस्ता replaced by bone. कार्टिलेज वहाँ से रिजॉर्ब होना शुरू हो जाता है और वहाँ पे बोन डिपॉजिट होना शुरू हो जाती है. इस तरह के प्रोसेस ऑफ बोन फॉर्मेशन को, जहाँ पे पहले मीजेनकाइमल सेल्स कॉन्ड्रोब्लास्ट में कन्वर्ट होते हैं, वो वहाँ पे कार्टिलेज सिक्रीट करते हैं उनका, और फिर उस कार्टिलेज मॉडल की जगह बोन ले लेती है इवेंचुअली, या बोन सेल्स आके वहाँ पे बोन बनाना शुरू कर देते हैं. दिस टाइप ऑफ the bone formation is called the endochondral bone formation. This is it for today. Endochondral bone formation की डिटेल में हम नहीं जाएंगे. I am going to leave it for the next lecture. I am going to see you guys at 11 o'clock Tuesday inshallah for the question and answer session of this lecture. Allah Hafiz.